life science course just to the end of the second year. So it's specialty the soil fauna. So today I give the time to I think he will introduce himself. So I would say welcome
Thanks, Zach, for the very nice introduction, and thank you for having me here. Today I would like to share with you basically a journey that I have been taking for about 9 years. And back then I actually had been working on earthworm for about 10 years, going through topics about taxonomy, systematics, phylogeny, and biogeography. But there was also a point when I started to turn myself from an earthworm specialist into a soil ecologist and try to understand the linkage between soil fauna and ecosystem functions. And of course, there's a reason behind this, and I'm hoping that throughout my talk, I will be able to explain to you the context. So I know everyone is very busy eating, but I would like everyone to give you one of your hands. So there are 7 pictures here, and how many of these do you think are soil animals? 7, please raise your hand. 6, okay, and then 6, 5. Right, so you guys can notice that I actually lower my arm lower, right? So you guys did a pretty good job. There are 7 animals here that are actually soil animals. So for instance, say beetles. Its larvae actually need to live in a soil that has a lot of humans. But I think among these, the most striking one would be cicada, especially this picture is this cicada with the red eye. It's actually the 17-year cicada, or called the periodical cicada. These animals, their larvae spend 17 years in the soil, feeding on xylem foliage of tree roots. And then after those 17 years, they emerge in late spring, about maybe late May or early June, when the soil temperature reaches a certain degree. And then they emerge in really large numbers, and then reproduce, and then die within just 2 to 4 weeks. So the density can actually reach about 300, 400 individuals per square meter, and they die pretty much together. And that means when they die, you have a huge flux of nutrients and organic material that can become nutrients and food for soil animals, plants, bacteria, and fungi. And so this definitely would have some impact on the ecosystem below ground. So the soil is actually a habitat for a very diverse group of organisms. You can have large animals such as earthworms or isopods, or some microscopic ones such as springtail, mites, and nematodes. And the numbers that I'm showing here are the estimated numbers of species in the world. So let's just take earthworms, for example. There are only about 6,000 described species of earthworms in the world right now, but this number you're seeing here is a lot higher. So with this diversity, the soil is sometimes called by some people as the poor man's tropical rainforest. Because in a square meter of soil, you can find hundreds of species of animals. If you go and basically count them, you can mostly find about 50,000 individuals of springtail and about 5 million individuals of nematodes. So this is a really diverse habitat with a lot of individuals. That means you don't really need to travel far away, and you can actually study sort of the diversity that's underneath your feet. But there's a problem. These animals are really, really tiny, so you need to find a way to actually get them out of the soil. And this is just an example that people use for microarthropods. This is called the birdie's funnel. Picture here is the setup that I had in my lab last year, kind of like the Christmas light, and Christmas is coming. So this is called birdie's funnel, and the way that it works is you basically heat your samples. You can put your samples here. It could be leaf litter, it could be soil. And you basically heat your sample with a heat source. Here is a light. And then the litter would dry out from top. And these animals that actually require soil moisture would move downwards through the funnel and then fall into a container that you can use to collect them. And of course, you can keep the animals alive, but you can also keep them in ethanol. And here's a picture of a setup that I had when I was back in the United States. And so these animals, of course, they look a lot nicer when they are alive. I'm just going to show you some examples, and I can go through this again. So these are some mice, and you can see some of the mice actually are pretty hairy. And remember, these are really small. These are like 1 millimeter, 2 millimeter individuals. There are mice that actually have camouflage on the back. There could be mice that have a shield on the side. These are the same individuals. And those shields are basically like protective gear, and they are actually movable. So these animals can really hide their legs underneath their body and close the shield, so basically form a bow. So there are also springtails. This is an individual feeding on fungi. And some of springtails, different body forms. Some are globular, some are much longer, pretty colorful, very nice looking. Again, these are animals that are about 5 millimeter long, 1 millimeter long. So really tiny. Some more springtails again. And so there are also these animals called pseudoscorpions. They look like scorpions, but they don't really have the tail. Again, and they are really, really small. And these are actually the sister group of scorpions. And so this really nice drawing sort of illustrates the complexity of the habitats that these soil animals are living in. So they are not just soil animals. This is a habitat where you can also find plant roots. You can also find bacteria. You can also find fungi. And these animals or these organisms below ground actually receive their nutrients or food resources from above ground in the form of animal carcasses or poop, root exudates, which are secreted from the roots, or leaf litter, or woody debris. And so with that, I actually have talked a lot about this diversity that's in the soil, but soil is important in several other aspects. I'm going to go through this in a minute. So when we talk about energy flow in an ecosystem, we usually look at how energy goes from the primary producer into the greater system and from one trophic level to the next. But if you just open your ecology textbook, and you will notice that the majority of the primary production actually becomes the dead organic matter, which gets into the decomposer system and gets used by soil animals, bacteria, and fungi. And so the soil ecosystem is important not just because of nutrient cycling, but also because of the flow of energy. So I have to apologize for this really, really ugly graph, especially in a seminar like this, but I haven't been able to find a better one that actually explains what I will be getting into right now. And so this is a global carbon cycle, as everyone knows. And you can find here, the soil over here. If you look at these numbers, you can actually see that soil is the largest active terrestrial carbon pool. And there's one thing that's called soil respiration, which is basically CO2 released from soil. And soil respiration is actually equivalent or even larger than plant respiration. And those two together is offset by what everyone knows is photosynthesis. And that leads to a net carbon fixation of 1.4 gigatons per year into the terrestrial ecosystem. Of course, this graph is pretty old, so these numbers are probably changing, but the general concept is still the same. So basically, soil is a carbon, or the terrestrial ecosystem is a carbon sink. But as you can see these big numbers, if you have like a 3% increase in soil respiration, which is basically, again, CO2 released from soil, then you can actually turn the terrestrial ecosystem from a carbon sink into a carbon source, a source that actually releases CO2 into the atmosphere. And so, how do soil animals affect the release of CO2 into the atmosphere? Well, the simple answer is they affect the soil microbial community. Because the soil microbial community is the primary source that decomposes soil organic matter, which after decomposition becomes CO2 and gets released into the atmosphere. And so the soil animal can affect ecosystem function, primarily CO2 emission from soil by affecting the soil microbial community. And this is the general concept that I will be going through again and again today. And so I will, today what I will be doing is I will present a case study about how soil animals affect CO2 emitted from soil by using a case study focusing on earthworms. So I'm just going to give you a brief introduction about earthworms. So there are several pictures of earthworms, and they can actually come in very different colors. You have red ones, pink ones, some green ones, and some very general brown ones, and something that looks kind of like purple. You can also have some very colorful species, like the one down here is a species from Australia, and one up here is a species called Archiphylloctima militans from the Philippines. This worm right over here is about 30 centimeters long, so it's a really big earthworm living on the forest floor in the tropical rainforest in northern Philippines. So look at these patterns on the body. Does that remind any of you anything? They look like fried egg. And so a lot of people actually call this earthworm fried egg earthworm. And what's interesting about this worm is there are several species that sort of have this black background with the white dots, but this is the only species with the yellow dot. And there are species that are really, really long. On the left is a species from the Mekong Delta, Mekong River in Thailand. That's a species called Amethus megongianus. That species can reach about 3 meters long. On the right are some species from Ecuador in South America. Well, you can see how big they are and how big the eggs of these worms actually are. So, before I get into the science part, I'm just gonna get one question out of the way since I've been asked several times by people inside and outside different seminars. Are worms edible? Yeah, of course they're edible, right? Because there's a lot of proteins. This is a snapshot from a video that was taken in the 1960s from Peru. You can see the native people cutting a giant worm, clean the gut, and then throw the meaty part into a hot pot. You can see the meat actually curled. And so they're edible. And you'll be saying, hey, Chih-Han, come on, this is like, what, 80 years? No, sorry, 60 years in the jungles. No modern human beings would actually eat these type of things. Well, this is an Amazon website, and I can remind you, today is actually Black Friday, so go shopping. All right, so I bought this last year, and I actually still have a bag in my office. Yes, I did. I had 2 bags, and it's kind of early. Anyway, so called earthworm jerky. It's not terribly expensive because people are actually trying to find alternative protein sources for human beings because people are concerned about all these carbon emission that we are producing because of cows and pigs and so on. So alternative protein sources. And you can read the label, no artificial flavoring, no artificial color, mono dehydrated, not fried, really, really healthy. Great source of protein and great gift. Alright, check out the review about Chih-Han. I'm gonna be giving these to someone that you actually know. Alright, so moving on. Everyone knows who this guy is, I'm pretty sure. That's the.
Excuse me, I have a question for you in the previous slide. So what kind of earthworm species is it?
Well, the name actually is Evolutionary SP, which means the purity mode earthworms. Well, name of the one that I ate, they look kind of big. So my guess would be they are Amethus sagittarius, but I don't really know.
So do they collect from the wild or they kind of raise?
I don't know, probably. This is from Thailand. So I can dig into that a little bit deeper. I don't really know, but and also it says Evolutionary SP, which means something because I'll get into that same thing later. It belongs to the Evolutionary group, which actually is a group with like 13 genera and more than 1,000 described species. So we don't really know what they are. So that's the outgoing president of the United States, right? So next year he's not going to be the president, no one's going to remember him. So I think I've been using this cartoon for about 3 or 4 times. So I think this might be my last chance to use it. So I'm just going to take it. Okay, this campaign slogan was "Make America Great Again." But I would like to argue that America has been the greatest country in the world for the last 20 years to study earthworms. And the reason wasn't because it has a really high earthworm diversity. Well, do the math, there are about 180 species here compared to 120 species that we have in Taiwan. And this is a much larger country, so 180 is really not a lot. But the thing is, they have a lot of non-native or invasive earthworms in the United States. Many species from Europe, many species from Asia. And so in the last 20 years, the US government, or actually the National Science Foundation, devoted a lot of funding to study invasive earthworms. And that's why it had been the greatest place to study earthworm in the world for the last 20 years or so. But it's kind of dried out. This is a picture that I took about 2 weeks ago in Fushan Botanical Garden, Fushan 植物园. You see these are basically deer fences. They are keeping the barking deer, Chang, away from eating the plant inside. And so you can see the very dramatic difference between the plants, abundant plants that's inside and not so many individuals that's outside. But these fences can only keep away animals or herbivores from above ground. They won't be able to keep anything that goes in from below ground. And these are pictures from American forest that's in the United States, one that had been invaded by earthworms on the left, one that had no earthworms on the right. And you can see a dramatic difference with lots of seedlings that's on the right, and not so many on the left. Because the worms actually, some of the species are actually herbivores that would actually eat the germinating seeds or the seedlings. And so those effects can actually be seen when you even dig into the soil. On the right is a forest with no earthworms. Again, these are American forests. You have the leaf litter layer on the top, and then the organic soil, mineral soil. Really well layered. You can see this really thick litter over here, and the soil that's much lighter underneath than the soil that's on top of it. But when the forest was invaded by worms, the worms eat all the leaf litter, and then they move into the soil, they translocate all that litter into the soil, and give you a really, really homogenized soil profile. And so, what does that have to do with carbon? The worms can move the carbon, the organic material, into the soil, which, as I mentioned earlier, can be used by the soil microbes. And so there could potentially be some changes in soil respiration. And so, of course, people, as I said, there was a lot of funding in the last 20 years, and people have been studying topics like this for a long time. And this is a really, really nice review paper published in Nature Climate Change in 2013. And the title reads, Greenhouse Gas Emissions from Soils Increased by Earthworms. And then it basically was a meta-analysis paper that summarized data that had been published before that year. And you can see for CO2, the effect size of earthworm was somewhere between 22 and 40%. And for another greenhouse gas, N2O, that range was much larger. But the bottom line is, okay, earthworms increase soil respiration. Earthworms increase the release of greenhouse gas from soil. But if you break that down into different components, I'm not going to go through the table, I'm just going to let you read the one that I highlighted. There's experimental periods. There are short experiments, intermediate and long-term experiments that lasted for more than 200 days. And what you're seeing here is for short-term experiments, you do have a significant effect on CO2 emission as well as N2O efflux. But for long-term experiments, there were no data on N2O, and there were only 19 studies that lead to a 12% increase that was not significant. And so if you're thinking about something global, we're thinking about something that's related to climate change, we're not looking at 30 days or 30 to 200 days. We're looking at a year, 10 years, and 20 years. And so that basically is saying the conclusion that many of the earthworm people have been talking about actually kind of premature and not really very well supported by the data we actually have at this moment. And so, as I mentioned, there were different groups of earthworms, invasive earthworms in the United States. And we can actually put together a timeline about when they actually invaded. So before European settlement, since invasive species is something that human beings make, so before European settlement, there were only native species. And then in the last 300, 400 years, the European species came in and either replaced or coexisted with the native species. And then in the last 100 years or so, the Asian earthworms went in and became the second wave of earthworm invasion. And these Asian earthworms only started to show up in forests that actually got people's attention about 2010. So only 10 to 15 years ago, that was the timing when these Asian earthworms were widely noticed by people living in the United States. And so these Asian earthworms, the question I just got earlier, these Asian earthworms actually belong to the phyllotimoid earthworm complex. And they originally from East and Southeast Asia. They basically include most of the species in Japan, most of the species in Taiwan. And they are now for genera in the United States. And among all these 16 species, there are 2 species that actually concern people the most. They are Metaphori and Amethus aggressivis. I'm reading this a little bit slowly because I will be reading these names several times throughout the rest of my talk. The species are from Japan. They are really large. You can if you look at the picture here, this is a ruder that's about 15 centimeters. So the worm is actually pretty big. These worms are parthenogenetic. That means they can reproduce by themselves. They don't need to mate. They can reach really high abundance, about 150 individuals per square meter. So just look at 150 individuals of 15 centimeter long worm. That's around your feet. These worms are interesting in another way that they are actually annual. They overwinter as cocoons. And so what it means is the worms would actually emerge from the cocoons in late April and May. They grow really fast, become adults, reproduce throughout the summer, and then they would die by the end of fall. And so they would survive the winter as cocoons in the soil. You wouldn't be able to find any single individual in the soil throughout the winter. And they can grow really fast, basically 15 times of their original weight, going all the way from 0.1 grain in April to 1.5 grain in August. So that's a newbie, and that would be an adult worm. And they have been found everywhere in northeastern United States. This is a map, basically all the different colored dots of where they have been found. And you could have data that come from research. You could also have citizen science project data like IMAP and iNaturalist. But the bottom line is they are basically everywhere. And of course, they also would make the news since they are everywhere and being found by people. This is 2013 in New York, 2014 in Madison, Wisconsin, and then that's 2015 in Chicago. And we can go on and on with new reports every year, but one thing that people are wondering is, are these Asian worms competing or even out-competing the European species that have been in the US for a long time? And so one of the site that these worms were found was the Arboretum of the University of Wisconsin-Madison. And so a team in the university started to look at one of the plot in the Arboretum, and what they found was they took samples in both 2015 and 2016. And what they found was the Asian worm seemed to be expanding from the left-hand side of the plot into the right-hand side of the plot. And 2 really common species of European earthworms seem to be disappearing from the plot. You can compare the 2 different graphs. And so if they are competing, if the Asian worms can potentially outcompete these European worms, what do they compete for? And so one of the guesses that everyone would have is, space or food. Okay, so if they are competing for food, what are they competing? Worms eat leaf litter, so they can potentially compete for leaf litter. And that's probably true because both of these species are what we call the epigeic species. That means these worms that live in the surface and feed on the leaf litter, use leaf litter as their primary food resources. And so we basically have 2 players, Asian worm, Metaphori, European worm, Lumbricus rubellus. And so we can set up some sort of experimental competitions and then just look at the worm competing for leaf litter. So you can have a pot that has soil, has worms, and has leaf litter on the soil surface. But then the thing is, how do we know if they are competing for leaf litter? You can't really see what the worms are eating. You can't really see they are fighting or competing. You can't really see that they consume the litter. And so we can find some tracers in the food. And one thing that we can use is to use the stable isotopes. So carbon and nitrogen stable isotopes, carbon-13 and nitrogen-15. We can have nature-abundant soil, and then really enriched leaf litter that has a much higher content of carbon-13, a much higher content of nitrogen-15. Compare those two different numbers. And we can create these type of enriched leaf litter by feeding plants with 13C enriched CO2 and nitrogen-15 enriched ammonium nitrate. And so basically, you feed the plants and then you collect the leaf litter for the experiment. And so a higher value in the plot, so I'm plotting 13C on the x-axis and 15N on the y-axis. So a higher value would actually mean the animals eat relatively more leaf litter because the leaf litter was enriched in carbon-13 and nitrogen-15. And the lower value down here would mean those animals eat relatively more soil. And so for competition, we can have two different scenarios. There's one that the two compete, one that the two don't. For one, there is no competition. You can have two species that have different stable isotope signatures because they feed on different food resources. And then you put the two species together, nothing would happen. In that second scenario, there's competition. You have two species that feed on the same food resource, so they would actually have the same stable isotope signature. And you put the two species together, there's going to be competition, and there's going to be some sort of the isotopic niche shift that you would potentially see between the two species. And so now we have our plot, we have our mesocosm experiment, and we can look at the data. And the winner is the Asian worm. So on the left is the Asian worm, on the right is the European species, Numbricus rubellus. And again, 13C on the S, 15N on the Y. So these are data points when these species are alone. And when you put in the other species, you see the change in the data. The values increase in the Asian worm metaphoric higidophy, decrease in Numbricus rubellus, the European earthworm. And so there is competition, and the Asian worm appear to be a superior competitor. But that has not answered our question. So we now know that, all right, the Asian worm actually in some way replace the European worms. We now have some lab data, as well as field observations that kind of support that. But how about soil respiration? And so I showed this graph earlier about how these worms may affect the distribution of organic materials in the soil and carbon in the soil as well. And you can actually just go out, dig out the soil, and measure the carbon content in those different soils. And you can see, and of course you would expect to see something like this, a really big difference when there are earthworms compared when there are no earthworms compared with when there are earthworms in the soil. So a very different soil carbon profile when you actually measure those values. And so that translocation of carbon in the soil, as well as the organic materials in the soil, is going to change the food resources that's available to the soil microbial community. And so we can have a conceptual diagram here. The food resource availability is going to increase soil microbial biomass. So there's more food, there's more microbes. And that increase in soil microbial biomass would increase the energy that's being required for maintenance, for growth, and for reproduction. And so the microbes will consume more soil organic matter and eventually lead to more loss of carbon through soil respiration. And so potentially, this is the way that many of the soil organisms, or a more simplified way that many of the soil animals can actually affect soil respiration. And so now we have our conceptual diagram, we can have some sort of predictions. So I'm plotting the data points that have no earthworms. And then if you have earthworm, you would expect to see a higher litter translocation, a higher microbial biomass, and a higher soil respiration. So pretty straightforward from the previous diagram. And today I'm just going to talk about the microbial biomass part. I have to clarify that the soil microbial biomass data you're seeing in this coming slide is coming from the mesocosm experiment in which the competition experiment was taking place. So here's the data. And again, try to simplify things. We're going to be looking at only control and the treatment with metaphoric Higgins-Dophy. So that's the red arrow are the ones with the earthworm, and the gray ones are the one just the control that has no earthworms. As you're seeing here, so we can use phospholipid fatty acid as a biomarker or indicator for microbial biomass. For total bacteria, gram-positive bacteria, and gram-negative bacteria, we're seeing a negative effect. So these Asian earthworms reduced microbial biomass in the experiment. And that result was consistent with a paper that was published in 2010 in Ecology, basically again showing decrease in microbial biomass under the treatment of another Asian species. So I mentioned metaphoric Higgins-Dophy and Amytas agrestis, and this is the other species that's being invaded. And so that looks really nice, right? So you have your prediction, and then, which basically says the worm would increase microbial biomass. And then you have your data that says, well, no, the prediction is wrong, which is, oops, not so good. But you have some other published data that actually show the same thing. So for any graduate student or anyone, this kind of is like a pretty good scenario because you can write a lot in your discussion and you can just say other things why the theory or why the prediction was wrong. But back then I happened to have a set of field experiment going on that lasted for about 450 days. The experiment was not designed to measure either soil respiration or soil microbial biomass, but it did have the right setup to do that. So I actually had soil that was in the freezer, so I took out those soil and did the same analysis. And I was seeing the opposite effect. There was increase in microbial biomass in the field experiment, which was consistent with the prediction and was totally inconsistent with the lab experiment. And so what this was saying is, well, which one do we want to believe? I think we would want to believe one that has actually lasted for more than 400 days. So the lab studies were probably wrong. Those were probably some artifact that was introduced during a 30-day or 60-day or 90-day short-term experiment with soil that was heavily disturbed as well as being contained in a small container like a bucket and so on. And so the truth is, we really know nothing. We really know very little about all these systems. And there are other factors that's in play. One is phenology. So what you were seeing was a bar graph that I presented. That means that was a one-time sampling. But things happen throughout the whole year. So this is earthworm activity that I sort of plotted for European species. And as I mentioned earlier, one thing unique about these two Asian species is they are annual. So their activity would actually be super high in summer, but pretty much nothing throughout the winter. So they have very different activity patterns. They have very different phenologies. And so this is the point when I took the sample in the field for that 450-day experiment. That was somewhere in the fall, before all the worms died out. And so that was just one-time sampling. So what was happening throughout the whole summer? We don't know. We really don't. And so the bottom line is, I think phenology is really important, and there really need to be some long-term study to help us looking at a system like that. And so going back to the study that was done in the University of Wisconsin-Madison Arboretum. So this is a team that collected samples in both 2015 and then 2016. And another team led by one of my collaborators started to collect sample in 2017, so 1 year after the original sample collection. And they found that some of the sites that didn't have worms in 2017 now have worms in 2006, some of the sites that had no earthworms in the previous year started to have earthworms in 2017. And so they categorized the sites into all sites, which were sites that had worms before 2016, and new sites which only show earthworms showed up in 2017. And then they did some microbial analyses as well as soil respiration analysis. And so on the right-hand side is the 16S metagenomic data for soil microbes. Really nice data. You see separation between the new sites and old sites. That's microbial community. We don't really know what groups are in there. And then they also did a in vitro CO2 flux analysis that basically had significant effect that would show there is higher CO2 flux in those old sites. And so that appears to be saying that the worms actually increase CO2 flux from the soil. But I wrote down in vitro, of course, not for no reason. It's an in vitro analysis, which basically means you collect a small amount of soil, you put those soils in basically a test tube, and then you observe CO2 flux from the test tube for about 6 hours. And then that's the CO2 flux. And so it really isn't what's happening in the field. It's really, really heavy disturbed soil. That the disturbance happen when you took the soil, when you mix the soil, when you put the soil into your vial. And so that basically gives you some potential of CO2 flux, but that doesn't necessarily mean that it's the CO2 flux in the field. And so I started to wonder if we, myself included, have been asking the right question, but trying to answer that with the wrong approach. And because these are really nice data, and I would love to have 16S metagenomics data when I were in the US, but we didn't really have funding to do something like that. But this approach really help us answer the question that we want to answer, the thing that we really, really would like to know. And so how do you actually measure soil respiration? You have to actually install a ring in the soil, and then you go out, and then that ring stabilizes. And then you go out every week, every day, every week, or every other week. I would say every other week is probably the minimum that you can do. You probably would prefer to do that every week. You go to the field using instrument to actually take samples. And so with the variability in nature, you need a lot of samples. And as I mentioned, you need to do these long-term to actually help answer the question. So coming back to this study, it's a really nice study, easy to understand, but it gives people an impression that the question has been answered. But I think we really don't know what the answer is. But with a paper like this published in a really high-profile journal by some very well-known scientists, it's getting harder and harder now to argue that, well, there needs to be more research devoted into study that. And so just one thing from my personal experience, a lab mate of mine was actually measuring soil respiration under some earthworm removal treatment in the US for 2 consecutive years. She went out every other week, initially every week, but then every other week to collect CO2 flux sample. And the data had not been published, and I guess you guys probably know why, especially if you are a faculty or graduate student. The results are not significant, so the data sit in a drawer, in your desk, in your laptop. It's not significant, so you're not publishing the data because they don't look interesting. And so the data that got published were the ones that look interesting, which are the ones that show earthworm increased soil respiration. I'm going a little bit faster than I expected, but I think that's kind of nice. So I'm just going to use the following several slides to wrap up my talk. You've seen this drawing before, but I just want to emphasize that this is a really complex system, and we actually know very, very little about that system. And I have been talking about diversity, and people are not really aware of the diversity that's in the system. But in addition to species diversity, there are also functional diversity that's in the system that we really don't pay attention to. For instance, these are earth-moon functional groups. There are 3 major groups and some subdivisions as well. And by using stable isotopes, again, 13C and nitrogen, carbon-13 and nitrogen-15, you can actually see more feeding groups than the functional groups that I'm indicating over here. So there are actually more feeding groups than the functional group that people are seeing. And and how about some worms that are so big? This is the metaphyre taiwanensis, an endemic species from southern Taiwan. It's a big worm. And it's quite abundant, actually. You can find them pretty easily in Kaohsiung and in Pingtung. And so that big worm is definitely part of the anisic earthworm group, which are species that have permanent vertical burrows going really deep in the soil. For species in Taiwan, could probably be 30, 50 centimeter deep. And then they come to the soil surface to feed on the leaf litter on the surface. And we really don't know much about that. And we don't really know how they can actually translocate mineral soil from the bottom, as well as translocate organic materials from the top into the soil. And how about other animals? How about mice? Most people, or most of the literatures, would be saying that Oribetic mice are fungal feeders. And this is a paper published about 9 years ago, basically saying, well, there are a lot more feeding groups in Oribetic mice. You can even have predatory or scavengers that feed basically on animals. And there are different groups of decomposers as well. And also springtails. There are a lot more feeding groups in springtails, which is also a group that traditionally people think they are just fungal feeders. They're all more or less the same, but no, they're not. And so my take-home message really is pretty simple and echoes the title. We can't really say that invasive earthworms increase soil exploration, even though there seems to be evidence for that, even though I personally kind of believe that as well, but I don't think the data is strong enough for us to actually say that's the conclusion. We also know very little about the linkage between soil organisms and ecosystem functions. With that, I would like to acknowledge my funding sources, which are ones that really keep me going for the last 10 years. Also my collaborators, many of them are still ongoing. And also my lovely students who are probably watching me or laughing at me in front of the screen right now in the lab. And thank you for having me.
Thank you for Chih-Han to give us the opportunity to see that really unknown world of the Earth ecosystem. Any questions?
You go through PDA or 50, where you show the competition of two earthworms.
Yes. This one?
Right. So are they already different? I mean, in the isotopes, are they already different?
These two values originally?
Right, because it seems they have separate part already.
I don't know. But one of the things is, so if you imagine how an experiment would be done, these are worms that we collect from the field. Right? We wouldn't be actually raising the worms from cocoon, we wouldn't be feeding them with the same type of soil and litter. So these are worms that we collected from the field, pre-incubated in the lab for about a month, and then put them into the experiment. So that original difference may come from the different habitat that they showed up in the field. Not necessarily the food difference that they have in the field. Because these worms are actually collected from the same side. So it's sort of something that we can't really deal with, just the way it is. But that's a very good question. I can actually show one thing. I don't really have the slide. So the graph on the left is from more or less a different study. So that epigenic species is actually the European worm that was in the experiment. And from that same location, we were able to collect the same Asian earthworm. Actually plotted the data in a different graph. And so because this is data from 1 year, and the data that I'm talking about is the data from another year. So I didn't really plot them in the same graph. But in that graph, you would have the European worm somewhere here, and then the Asian worm basically cover the whole ellipsis of the European worm. So they are, in the field, their isotopic niches do overlap. And that Asian worm actually has a much wider isotopic niche compared to the European species. I should include that slide in my next talk, I guess. Thanks. Yes. Pardon? Yeah, it's possible. I have not heard about that. I think study competition for long run is really hard, especially for when you are focusing on these animals. When I was working on that project, I couldn't find any relatively recent studies focusing on earthworm competition. I think the one that I was able to find was almost 10 years old, because there was just no approach for you to actually observe, record, monitor, or quantify the things that you want to measure. And so I guess that's a pretty good question, especially when I was restricting the worms in a small container. So there's definitely space competition as well, but I can't really tease that apart. Very good question. Thanks.
Can you show me the result of your 450 days treatment?
Sorry, could you speak louder?
Oh, can you show me the result of your 450 days?
Oh yeah. The microbial data, please. Yes.
Because your final result is not consistent with the laboratory experiment. But have you measured these bacterial biomass like every 30 days or?
In a field?
Yes, I mean.
No, I didn't. And that's the thing. I had only one data point.
Yeah, that's the question I want to ask. Why do you just have one?
So the experiment was actually designed to look at soil microalgae as well as nematodes.
Okay.
And because I mentioned earlier, I have a laminate measuring soil restoration for 2 consecutive years, and I see the effort that she had to put in to do that. And so I thought, well, this seems to be a settled question. There's effect on soil respiration by earthworms, so I didn't think about measuring soil respiration back then. So basically it was a 2-year experiment. For these animal species, they die at the end of the year. So at the beginning of the second year, we put in individuals, and then using electroshocking to monitor the other species that we put in. And then so I sort of just wanted to have an end-of-year sampling, which was actually planned to be at the end of summer. But remember, the U.S. government had several years of government shutdown at the end of the fiscal year. I was there. I took samples from one of my replicate, and then the whole institution, the Smithsonian Institution, shut it down for 2 weeks. Yes. No one goes in, no one comes out. Just no one goes. So there was no way for me to. Well, yeah, but that was a pretty good question. So.
In my opinion, I think maybe the biomass of this bacteria that will be like a series of changes
Yes.
The first 20 days or 60 days they will decrease like in the regulatory unit, but maybe for long term it will change.
Yeah, it's totally possible. And so what I think, so if I were to go back to do the experiment or to go to the other people's side to take samples, I would definitely do that every month
throughout April to, let's say, September. And just to basically have an idea about how these values changes. And because, again, these are one-time sampling. And I even only saw that they wrote the samples were taken in summer, period. I didn't even know whether that means June, July, or August. So there are, well, it's field work, so there's a lot of effort that people need to put in to actually collect samples, to process samples in the lab, to actually measure everything. And so I understand that if your focus is sitting as metagenomics and you have just a little bit of money to do that, you probably wouldn't want to have 100 samples to analyze. But again, whatever they did or whatever I did didn't really answer the question. It's really not the right approach for the question. And I think what really needs to be done at this moment is someone can actually do field sampling for 2 or 3 years. Field monitoring plus maybe a 2-year or 3-year field experiment with slightly larger plots. But it was really a lot of effort. As you saw, the plots that I had, they were not very big. This is the metal wested, what's it called? West basket. So it's about 5 gallons. So it's like a big container. It's not really very big. And so to do that, to start the experiment, we had to dig out the soil about 30 centimeters deep, somewhere between 20 and 30, and then separate the soil so visually separate the soil into the darker organic soil and the mineral soil in the bottom, and then really homogenize the soil. That's what you would imagine to have an experiment that you can actually compare one treatment to another. So really homogenize the soil, sieve the soil through a 4 millimeter sieve in the field. And so that was a lot of work. And so imagine a team needs to do something like this with a slightly larger size of their replicate. And yeah, but I think that's doable. And that's doable for a team responding. And I think that really is something that needs to be done to actually address the question. And that's just one data point. It's like just one experiment, and it doesn't really say that you can actually apply the result from that one into like the word.
Thanks for the talk. So it seems that the main opinion or the main findings in the literature is that people think that earthworms is the food sources for microbes. So then that enhances the biomass in the growth of microbes and then improves the soil restoration. But I'm interested in the links between microbes and earthworms. Are there other mechanisms or cases that you know that actually the earthworms can make some physical or chemical changes in the soils, or changes some biological conditions that actually disrupts microbes or decrease the biomass or growth of microbes?
Very good question. I don't know. I can't even say I'm not aware of, because I don't think I go through some of the details of the infrastructure enough. But I do want to say a general thing about earthworms effect on chemistry and physics of soil is they would increase soil pore density. And they would also increase soil pH as well, usually slightly. And most of the time, the calcium content in the soil. So SOBs would affect soil microbes. But whether it's a positive effect or a negative effect,
I think we need a soil microbial ecologist to answer that question.
The behavior of the earthworm affected by other species, like the iguana in the
I don't think people care about earthworm enough to actually do research on it.
in the superficial layer will be more into the deeper depth because of the taiwan. I'm not sure for the data itself.
I don't know. I need to look at the data, but I want to say, well, I want to say if an organism that's large enough that actually disturbs the soil, the worms would definitely respond. I think small mammals yes. So there's a study on some small mammals, I want to say it's mole or something, that would actually change the behavior of worms because of the vibration that the animals produce in the soil, because they feed on worms. Because of the vibration that they produce in the soil, the worms would actually escape. And the way the worms escape is to come out of the soil. Come out of the soil to run away from the predator. But I want to say that's the only case I'm aware of. But also that's like when the worms actually already run into the predator. And so of course if they are predatory birds that feed on worms, as long as some of these ancient worms can actually jump. I mean we poach out, but it's just like thrashing and trying to escape. And that behavior would most often disturb the predators. So when birds try to catch worms, like American robin in the US, when they try to catch worms, these Asian worms would actually kind of jump a little bit. But that's like when they actually go into each other. So I don't know on a habitat scale whether these predators or other groups of animals would actually affect the behavior or habitat use of worms. Just not that many studies around.
So the invasive Asian worm are animals, and I wonder, what's the ratio, like how many worms are actually animals, and do animals often compete with those that are not animals?
When you say how many species are animals, do you mean like in the US or in general? Or
both, I guess.
In the US, probably 4 or 5 species, all those are Asian. In general, we don't know. Like in Taiwan, as I mentioned earlier, we have close to 120 urban species in Taiwan. So far, only one is known to be animal. But that doesn't mean that the others are not, because we don't just necessarily. We have data about where they were collected in the field. We have data more or less about the diversity, but that's pretty much it.
I was wondering if earthworm enhance the soil respiration because it has a large body size relative to other underground organisms.
Yes and no. So yes, they are big, so they would actually respire a lot, and of course there would be CO2 increase caused by earthworms. But the increase that they cause is really not big enough compared to the overall size of the soil respiration. So basically, most of the time soil animals, whether you think there are lots of them or a high biomass of them, the respiration of the CO2 they produce is really just nothing compared to microbial respiration. Or even root respiration would actually probably be higher than respiration coming from the animals. So probably would be an order or 2 orders of magnitude higher from the soil microbes.
To say that it doesn't matter what size of earthworm is, as long as if they exist in the ecosystem, they kind of promote the microbes and then that increase.
Yeah, it also depends on the biomass. Because you can have, just 1 mole per square meter versus 100 per square meter, or for some species that are a little bit larger than these Asian worms in Northern Europe. I want to say in Finland, they have recorded density about 500 millikilos per square meter. I don't even know how those big worms live in such a close place. So, 2 gram in the videos that are 2 gram in size, close to 20 centimeter long, and like 500 millimeters per square meter. I don't know how to reach that, but there are smaller ones that can reach like 800 millimeters per square meter. So I do think the total biomass differs a lot. If that answers your question.
Yeah, I'm just wondering, if it's possible to scale up from your field experiments. For example, here it seems like you can use the experiment to tell with earthworm versus without earthworm in different restoration. But then when I think of, for example, different locations in the U.S. or different other terrestrial ecosystems, and then it's difficult to expect whether there will be high or low.
Yeah, exactly. So we're doing any global carbon modeling. One component is for restoration, but whether a site has been invaded by earthworms. Well, I say invaded, that basically means they were literally means invasive earthworms. So we have lots of earthworms in Taiwan that are native. So I guess it's really hard to do so. I guess we just don't know how the different groups of invasive earthworms can have the effect they may have on different ecosystems. So it's pretty hard to scale up from any of the research that I've done so far, especially most of the research I've done were done in temperate forest. But it's a big goal. Yes.
Just out of curiosity, how many invasive earthworms are there?
It's always the hardest question for me to answer. And the reason for that is because we have a lot of sort of the naturalized species that probably have been in Taiwan for, 400 years or so. And it's pretty hard to decide whether I want to call that as an invasive species. I want to say we have about 30 to 40 non-native species or cosmopolitan species in general, and about half of them are definitely non-native, which excluded the naturalized species. And there are probably only 2 or 3 species that are really, really invading at this moment. Well, that's the data we have, so I maybe just know nothing because there were not a lot of data around.
Very interesting presentation. I'm interested in the link between the sea and the effect of earthworms on microbial restoration, because you use the quantity of lipids you can find in the soil to measure the biomass. But if you could do anything, how would you measure the actual biomass and the restoration of microbial communities in the soil itself? Because it's a challenge for microbiologists to measure restoration of microbes. So how would you do it?
Microbial biomass and microbial restoration.
I'm not a microbial ecologist. It's pretty hard. So people are, very happy to look at any of the 16S metagenomics data nowadays. And people would even compare the community structure, who is here, who is there, which groups are more dominant than the others. But I just learned recently that when you change the primer set that you use, you'll be actually seeing potentially very different results. So if you're using a set of primer pairs that amplify the B3, B4 region versus another one that uses a B4 primer, you would say, okay, great, the B3, B4 covers the B4. But actually what you're getting are going to be very different results. You may get more archaea using the B4 primers and others. And so, and that's why when I was asking around to try to figure out what kind of measures that I had for microbial biomass, I didn't really get something that was satisfying. And PLFA was what I ended up. PLFA was what I ended up had, partially because of the availability of instruments as well as something.
Some suggestion how to measure biomass.
These are the common ones, the way they're using the tests.
Yeah,
that's a common.
It's a common method, but it doesn't really give you Well, again, it relies on biomarkers, so it's relative biomass. I don't even feel comfortable comparing across different studies without using the same technique. And so I guess one thing is if you have a method that you can use to compare across different studies, would at least satisfy some of the standardization first, as well as to try to get actually an absolute amount. And then activity is another thing. I think that's even more challenging. Traditionally, for soil people, we have been using enzyme activity as a proxy for ecosystem or for function sets happening in the soil. And most of these enzymes are related to degradation of carbon resources, sometimes nitrogen resources as well. I'm not really very familiar with the details, but what I knew was, well, it's pretty tricky again, because those are extracellular enzymes that's in the soil, and they can sometimes be basically protected in the soil and then still functioning when you dissolve the soil in your 96 well plate, and then those enzymes are doing what they are supposed to do. But in the soil, those enzymes can actually be protected in some way that's actually making them not functioning in the soil. So extracellular enzyme is another tricky thing, and you can probably use respiration or substrate-induced respiration as a proxy for microbial activity, but then I guess it depends on the substrate you use. You probably would amplify some of the more dominant or optimistic microbial groups. So it's really just, again, those are ways that you can have direct comparison in your experiment, but I feel it's pretty hard to go beyond that to actually say anything about microbial activity.
Thanks for very interesting talk. So I found it very inspiring because I tried to do the same thing. I studied the soil in the ocean. So we also.
So you don't call it sediment?
I don't call it sediment.
These people who call soil in Mars will be like, well, there is no life in Mars, so that's not soil. Seriously, right?
So we also try to measure the respiration in terms of understanding how it's likely. So for us, because our soil is filled with water, so we can kind of separate the diffusive process from the total respiration. So we kind of use it as a proxy for microbial activity to measure the diffusive oxygen flux compared to the total flux, which in a similar way we're doing with a chamber. So my main study is about different habitats in the ocean. We know that they have different biomass and fauna. So similar to where the first one has effect on the respiration, I'm interested in different habitats with different amount of biomass, whether it has impact on the respiration. So what I found is there's no effect. Similar to but the thing is, what we found is that overall there's no effect. But we have looked at the contribution by the fauna and the contribution by the microbe. The actual difference is in the microbe. So what we found is overall there's no effect, but the relative contribution shows the difference. So I don't know if, because our soil is filled with water, so we can kind of separate it with different methods, but I don't know if in your study there's other way to measure the respiration to kind of understand the microbial processes.
Yeah, so for soil respiration measurements, I mean there are different ways that people can have use more remote sensing type of analysis to estimate, but for truly measuring, I guess the main major approach that people have been using. The first one is to
basically take the soil back to the lab and use substrate or not using substrate to induce the microbes in there to gives you a positive amount of potential. And then you get to the chamber measurement, as you just mentioned. And then there's another way called the gas wheel method, which you can basically have CO2 concentration probes in different depths of soil, and then use that concentration gradient or profile, try to basically model the flux. But the challenge for using that system is when you have a lot of water in the soil, when you have a lot of rain, when you try to do these in the non-temperate region, in the subtropical tropical region, you would usually get a lot of really messy results, because you actually, what happens is you can actually always have a lot of CO2 that's been trapped in the soil, not really releasing out of the soil, but then when there's rain, the rain will actually push the CO2 up and causing a flux. But that flux will also cause different concentrations in different depths, and sometimes the modeling result will actually give you a negative CO2 flux when you actually know that, well, no, that's what's actually, when you actually know that, well, no, there's no way that the flux was negative, that it was definitely positive and probably much higher than what it usually is. So that method is basically challenging, and I haven't seen many studies about how it has been improved or being implemented in the field. I know for NEOM, which stands for National Ecological Observatory Network, which is an NSF-funded long-term ecology monitoring network all across North America, from Alaska to Puerto Rico. NSF wanted to, so National Science Foundation is the equivalent of the Ministry of Science and Technology in Taiwan. And so the reason that NSF wanted to do that was because NASA had a lot of funding to do all sorts of long-term stuff, but NSF doesn't have anything. NSF is giving money away to researchers to do research, but it doesn't have its own project. And so I can remember it was probably like 4 or 5 years ago that NEOM network started to be functional. And what I was trying to say was they actually implemented both the chamber and gas well method for soil CO2 flux monitoring. So I guess we may be, since that's it's a cross of field sites with exactly the same instrumentation, having the same group of field technicians doing all the measurement, curating, cleaning, and analyzing the data. So we're hoping, I guess I'm hoping that within 3 or 4 years we'll see some data come out, actually use the data from both the chamber CO2 method, CO2 measurement method, as well as the gas well CO2 measurement method, and sort of being able to actually compare them and to know how good some of these methods are. But other than that, I haven't seen very good methods about soil respiration measurement, and the chamber measurement is the best one as far as I'm concerned, but it has its limitations. If you go out to measure CO2 soil respiration after a big rain, you'll see really high fluxes. And so the other challenge that people have about measuring CO2 flux is it's hard to capture those extreme events. And that becomes a problem. So that becomes a problem when you have many days when there is rain. And those are actually some of the fluxes that we are supposed to capture, but because we go out only on good sunny days to the field, we don't really capture those events. And so one way to do that is to have automatic chambers that are actually installed in the field, taking measurements every 5 to 10 minutes. And well, that's ideal. And the system expanded from the one in the picture here is actually equipped to do that. But so this is a system. So the system you're seeing here is something that you can carry to the field. But the system that I'm describing is from the same company, but actually has
robotic arms that would actually close and open a chamber in the field for you. And you have, wires supplying electricity to the instruments, which is waterproof, and then it gives you data for 2 years. But this thing over here is about close to $1 million NT dollars. And so imagine you want to have a setup like that in a field with, let's just say 3 different treatments and 10 replicates. That's a lot. So it's actually not very easy to do, and you also need to have infrastructures around to provide both maintenance and electricity. And also, if you're doing something like that in Taiwan, I guess they're gonna be gone in like a day or two. Someone is gonna seal it and just sell it to somewhere. And so it's just pretty hard to do anywhere in the world. Yes?
For soil restoration, for example, like improve the organic matters or loosen the soil or anything.
Could you say that again, sir?
I mean, do people use earthworms for soil restoration?
For soil restoration, yes. Yes.
Yeah, there are lots of studies in South America using earthworms for restoration after forest clearing and agriculture. Yes, and some of the studies actually find some of the invasive earthworms are pretty good at doing that. Yeah, because those are the only species that can endure the harsh environment. And those are the only ones that would actually survive. If you throw in the native species, they become nitrogen flux in the soil. They're dead. So for people in South America, I don't really have that problem because those non-native worms are already everywhere. And also, the one that they are using is from South America. It's just not native. It's a species native to a small part of Brazil, but it's found almost everywhere in the tropics now, Taiwan included. And so yeah, they're using that species because you can just go out and dig and you'll find eggs and worms. So probably for them, it probably doesn't matter whether it's an introduced species or not. Yeah, it's already everywhere.
I have a question comes back to your contrast experimental result. You show in some side you find an increase of bacteria biomass, in the other side you should decrease biomass. So what's the underlying meaning to the potential you can increase or decrease the
biomass? So to me, let me just go back to this one. Okay, so to me, it's pretty easy to imagine that worms would increase microbial biomass as long as the worms are actually incorporating more resources for the microbes. But on the other hand, the worms is a big animal. Its movement in the soil is actually disturbing the soil structure. And also some of the earthworm species would actually feed on the microbes as well. And so for that, the worms would actually decrease microbial biomass. And also for stuff like this, in a small container, you have worm and leaf litter, and I was seeing the worms were reducing microbial biomass. I think that was because the worms were using all the resources that was in the soil, and also the worms were just moving in that small container. There was nowhere for them to go, and so for the microbes, it's like you have a giant of something moving all over the place. So it's really not a very comfortable habitat if I were a microbe. So I guess that's probably my answer for why this is why I thought this was somewhat an artifact. And because you would, for a short-term experiment, people want to see effects. So what people would do is to look at the field density of the earthworm species that you would usually put in as many worms as you can. Of course, you have to be able to collect the worms, but usually people would just put in as many animals as they can in the treatment for a short-term experiment because you want to see significant results. So I guess that's my question. one funny, I think one thing that's related is there was a paper talking about intermediate disturbance hypothesis in the soil, looking at how earthworms may affect soil microbial community, and the conclusion was the intermediate hypothesis in the soil just didn't stand. But my thought or my comment on that was, well, whether a disturbance can be considered intermediate depends on the organisms that live in the habitat. And so in that study, earthworms is the source of the disturbance, and the microbes are what's in there. So for that experimental system, it was not really an intermediate disturbance. What was happening was not intermediate disturbance. It was more like an earthquake or typhoon.
I had some sort of share. So actually, when you talk about soil and talk about the sediments, but in water, it's actually the same. So if you take a bucket of water and then you measure respiration, more than 50% coming from bacteria. Actually, usually much more, right? So biological astronomer they study very hard to try to understand what's changed the respiration in the water, which is meaning how changed the bacteria growth rate or community mortality, right? So there are many physical or chemical mechanisms, predators, or virus infections. So I think I'll probably, according to the explanation when we were talking, I would say maybe biological astronomer is much more ahead in terms of that kind of knowledge. So maybe people that soil should read the biological papers and see how they make measurement and how they make the progress and what's the general understanding of the challenge. Instead of the event whale and then starting again, actually that how you get through study things like chemistry and our knowledge again, including molecular approach and how we do it. That's it.
Totally agree. I think soil ecology, soil ecologists like to call themselves interdisciplinary. Yes, it's a pretty tough research field because there are just too many things going on in the soil and you can't really understand every single thing. Usually you need at least a more sort of community approach soil ecologist plus a soil microbial ecologist plus a biogeochemist to actually do something that's meaningful regarding soil ecology research. But I think, yes, we really need to go beyond that and also, as you said, read literatures from other fields to actually tackle this problem. Yep.
Other questions? Thanks, Chih-Han. Okay, so I'll see you next month. We have a speaker actually over here that's here talking about microbial interaction. So it sounds like somehow for the whole semester we are talking about microbes. So I didn't put it to that, but I didn't kind of invite people in there. Okay, I'll see you next time. So you just be hungry and you food outside.